Hello, I'm Andrew Dufresne, and I just want to welcome you back to some AP Biology review. I hope your test prep has been going well on your own. And today we are going to talk about some molecular biology and genetics. If you remember last time, we talked about the chemistry of life. So we talked a lot about molecules coming together and building in complexity. And we ended with nucleic acids, and they're going to be our main star today. But first, I wanted to change it up a little bit, and I wanted to give you some context. So essentially, we're trying to figure out and try to explore how it is that we manage to have life pass on its information. What's connecting all of us? We're going to once again discuss some of the AP big ideas and skills of a scientist and everything, and I'll remind you about those. In fact, let me go ahead and resume this share, and then those are the ideas that I'm talking about here. So really, this idea of evolution or information storage and transmission, how are these actually coming to be? Like how does that actually happen from one generation to the next? And how did that happen on our planet? What's the history of that? So I thought I'd give you a little context. There were many hypotheses, of course, and we centered around one in the 1920s, postulated and put forth by Alexander Oparin, not Oparon, we'll talk about those later, and JBS Haldane, which is the most 1920s name I think I could ever come up with. They told us what they thought the early atmosphere likely was. Methane, ammonia, hydrogen gas, water. Notice that I didn't say oxygen, so that's going to be later on. But with that in mind, it then took us a good 30 years or so into the 1950s for Stanley Miller and Harold Urey to do their famous Miller-Urey experiment, where they put these gases together in a beaker, and then they lit some sparks. They provided that activation energy, if you recall from chemistry. This kind of mimicked lightning that would have actually been occurring on the planet as well, likely to a greater extent back then than even it is now, given all the chaos that was going on back then with things kind of trying to settle down. And sure enough, what they actually saw was that some molecules that resembled amino acids, also a macromolecule we talked about last week, started to form. And then the questions became, okay, well, if this can actually generate these molecules, and then we know that those can come together to make proteins, what is allowing all of the stuff that works, all of the proteins that are doing their job, and allowing us to increase in complexity? How is that information being passed down so that every generation doesn't have to reinvent the wheel? It's not as though we keep having an origin of life. It's just started and then continued from there. So that's what's going to be basically today's topic, is this molecular biology and the genetics, this idea of there being some kind of molecule that can encode for these amino acids, which then come together to make proteins. The going hypothesis on that was that RNA was the first thing. You may recall that being one of the nucleic acids. So we had over here from our sugar for DNA, our sugar for RNA, and then an example of a DNA base, because there is no hydroxyl group here. And remember, this was adenine. You don't need to know the structures. And the first thought was, in fact, that RNA may have started. RNA is nowhere near as stable, but it's a lot more flexible. It's single-stranded versus double-stranded like DNA. Again, we'll get into that a little bit. And so its ability to be flexible, and then the idea that it could come together and make some kind of replicable code, and then lead to the amino acids and proteins, that really seemed to make a lot of sense with everything that we knew. And then the question was, well, given the fact that it is so unstable, how would nature go about addressing that so that these things could stick around longer and be stored and grow in complexity before being passed on within a lifespan? And that would be where DNA enters the picture. So A little reminder again about these nitrogenous bases. This is going to be our literature of biology. These are going to form our codes and then tell us what we can get from those for amino acids. And then down here we have just a little schematic showing that one of these bases attaches to a sugar, again, deoxyribose for DNA, ribose for RNA, and then a phosphate gets put on. And this right here is a nucleotide. Now, you, like I said before, do not need to know the structures of adenine, guanine, uracil, thymine, cytosine, but you should know of those names, and then also A, G, U, T, C, oftentimes just A, G, T, and C, because those are actually going to be our DNA players, and then uracil comes in to replace thymine in RNA. Again, I'm expecting a lot of this to be review. If you have any questions, I want to remind you about our Q&A function. It worked pretty well last time. Go ahead and throw a question up there. There's also a raise hand situation in this Zoom webinar. So if you do want to alert me to the fact that there is a question, go ahead and raise your hand, and then I will know to check over there. Now, what else do we need to know about these nitrogenous bases? It helps to remember which ones are purines and which ones are pyrimidines. We don't really need to get into the chemistry of how those names came to be. Essentially, I like to go with cut the pie. The molecules that have a more circular style like this, as opposed to these lumpy, wonky ones that have a 5-atom situation coming off the side, these more circular ones kind of resemble a pie. And then sure enough, C, U, and T are the pyrimidines. So once you know those, you know the other two are purines. That's really all you're going to need to know about these, but they are going to keep coming up, like here. So something else we talked about last time, in addition to molecules coming together, monomers coming together to then form polymer chains, we talked about directionality or endedness. That's extremely important when we go through the molecular biology of everything that we're going to talk about today, because it's kind of like reading a book. Different cultures and different languages are read in different directions in different ways. In the Western world, English and a lot of European languages are read left to right, top to bottom. Hebrew and other Middle Eastern languages are read right to left. If you were to do that differently for each of those languages, nothing would make sense. Same thing with our genetic code. Now, was it set in stone in terms of reading it? Again, we're going to talk about this 5' to 3' aspect, but was that set in stone? No, it was a convention that we came upon and then continued. So much of this in biology is stuff that we observe and then we kind of go with something and then continue that on so that we can build upon past knowledge. It's not as though nature said you have to read it in this direction. So what the heck am I talking about in terms of directionality with these nucleic acids? Well, here we have on the left a strand of just an example of single-stranded DNA. You have here, there's no reason for this to be an X. I am doing what I can to find images because creating this in ChemDraw would be insane. Also, I think it's kind of nice to look at these old images. Kind of makes you feel like you're back in the 1950s. They're so old and the resolution is so poor, and it's like we're discovering everything for the first time ourselves. So we have here deoxyribose connected to our phosphate and then our base. And then here is the phosphate from the other base, the other nucleotide, and then it's set up, and then it continues on in that direction. Now, are there other phosphates coming off of this? This is kind of more the orientation. So this phosphate on this carbon is this phosphate coming off of that carbon. Now, and that just continues basically down the line here. So these are on the outside, the sugar phosphate backbone, and then the bases are here on the inside. And this right here, this is called a phosphodiester bond. Essentially, when you have an oxygen connecting two things, an ester bond, and then because this has a phosphorus in the middle, we call it a phospho, and then diester, two of these. A little more than you need to know there, but it's on the slide, and I wanted you to kind of understand that. And you will come across those terms when you do a review It's going to be a minor detail, if at all, in free response questions. We're going to talk about more about those later because things have changed since last week when College Board has provided some new information. Okay, so more about this directionality. You likely have heard already from class and from some of your studying, 5' to 3'. What the heck does that mean? Well, apart from it just being something that you want to remember, there's actually a reason. We number carbons in our organic molecules, and so that would be 1, 2, 3, 4, 5. And so this we call the 5' end, and this the 3' end. And sure enough, when you go in this direction, you're basically saying, okay, this 5' here is this base, and then here's that connection of the 3', and then I continue, oh, and that's 5', and then that's 3', and it continues on in that direction, as you can see on the right-hand side here. Now, this brings up something else that's going on. There's another chain here. I talked about how DNA is the more stable nucleic acid. That's because it's double-stranded. It takes some of the more reactive stuff going on, like the bases, and puts them on the inside. So that way they're less exposed. We talked last time about water and hydrophilic versus hydrophobic. It's not so much that these are hydrophobic, they are actually very polar and very reactive. This is a hydrophilic side here as well, also very polar with the oxygens. But we don't want our bases to be prone to activity and reaction with other things. It would be like writing something down and pouring water on the page and smudging the ink, or a candle falling on it and it's scorching the page, and apparently because we're in the 1950s, which also there was still electricity, just go with this. But we don't want these to change. We want that code to be preserved. So there's another strand on the other side. You'll notice that everything kind of looks upside down. It's not as though there is a top and bottom, kind of like the Earth. We always think northern and southern hemispheres. When you're out in space, it doesn't really make a difference. We just kind of go with a particular perspective. In this case, the other end is 5' down here, going towards 3' this way. We call that anti-parallel, where one is in one direction and then the other is in the opposite direction, but can be read were you to flip it in a similar fashion. Now, in the middle here are the bases, and those you may also recall, I'm not sure if you may have been taught about Chargaff and his rules, but they're about how the bases pair up with each other. So adenine with thymine and guanine with cytosine. Now, what is going on with these little guys here? Great question. Those are our old friend hydrogen bonds, or the intermolecular force in between them. So because these elements are polar, the nitrogen and the oxygen, and then the hydrogen's a little positive, while the nitrogen and oxygen are a little negative, those can interact, and that actually provides more stability. Imagine a ladder where you had rungs that had no connection in between. They were just basically kind of cut down the middle in the middle of the rung. That'd be a lot harder to kind of keep that together. But these hydrogen bonds are basically that factor that allows that rung in your ladder to stay stable so that the ladder actually stays together in one piece. So you are going to want to recognize DNA base pairing as well. We're talking about DNA and its stability and the base pairing and everything. There's a lot that happens for higher orders of life, like humans. We have about 20,000 genes in our genome, which means we'll get to what those are in a moment, but we have even more bases to make up all of those. So how do we get all of this stuff to be in a really tiny thing like a cell? Remember, I kind of want to bring you from really tiny stuff up to slightly bigger and bigger and bigger stuff. DNA packaging kind of does that too. So we have here at the bottom of the screen our sequence of bases. Here is our code. Here's the way in which we read it, and then that's going to tell us something. We're going to talk about how that tells us something as we move forward. But that also has what we call a double helix. That's that ladder coming together, that double strand, but then it twists like this. Now, I also want to point this out simply because I think it's super fun to point out random facts that only people that take this course are going to care about. But you may see a lot of drawings of the double helix, and it may look very neat where you have your strands kind of just interlock and go up straight like that. It's not that quick. It's not that neat. It's not that clear. So in fact, it has bends in it, and it has what we call a minor groove and a major groove. And those are just because of the way it twists. And I find that interesting because then what you can do is you can determine a little bit more about what's really going on here for the interactions between them. It's kind of like when we talked about the alpha helix before for a particular protein structure. There are different kinds of helices, A, B, Z, there's different kinds. But anyway, the helix then kind of twists and twists and twists. And then in order to organize that further, we put these proteins called histones. You don't need to know which ones, but it's a big honking molecule of about 8 different proteins. They get wrapped around on that. You may have heard the term beads on a string. The proteins, or histones, are the beads, and then the string would be the DNA. When those wrap around the histones, we call that a nucleosome. Again, there are plenty of other elements you may come across here, linker DNA and all this other kind of stuff in between. You don't really need to know about that, and I wanted to hit just the highlights, so we're going to move forward. But as those nucleosomes then get twisted and the DNA and protein get compacted and compacted and compacted, those also twist and supercoil, and then that forms chromatin. When DNA is wrapped around proteins like this, it's really hard for anything else to kind of get in there. Again, we're trying to basically store it unless we need it. And that DNA that is difficult to approach or to act on is called heterochromatin. It's inactive. It's not being expressed. Areas in between, or when they are more spread out and more relaxed, that's euchromatin. That DNA is accessible to other proteins, and it's active, and it leads to the expression of or allows for the expression of the genes on it. When that chromatin then condenses further, then we actually probably get to something a little more familiar, the chromosome. Given all the DNA that we have in our body and our genome, there are a bunch of chromosomes. The number of chromosomes differs between different species. For us, we have 23 pairs. We get 1 from our father and 1 from our mother. So we technically have 46 total. We're going to talk about this as we move into next week's class about haploid and diploid. So we'll be coming back to these chromosomes quite a bit, but I wanted to connect it to something that, again, was a little more familiar, I think. So these chromosomes are all kind of separated. We often think, oh, DNA must all be connected. Our entire genome is one bit. That's not the case. All of the DNA is in different chunks and fragments, and then that's in the nucleus. And it is often condensed down together into complexes. However, there are individual chromosomes, and that is usually the shape that we recognize. So what we've talked about so far are the nucleotides and how those come together. The nitrogenous base, A, T, G, C, or U in the case of RNA. We're going to talk about RNA more later. The 5-carbon sugar, deoxyribose in the case of DNA, ribose in the case of RNA. And then also the phosphate, purines versus pyrimidines, and then some structural traits. I mentioned the double helix and the anti-parallel strands. I didn't actually mention complementary base pairing. This is basically meaning that because only adenine only forms those hydrogen bonds with thymine and guanine with cytosine and vice versa. Were you to know the sequence on one strand, you would automatically be able to determine the sequence on the other strand, because wherever there was an A in that position, the other rung of that ladder, the other half of that rung of that ladder would be T, and then G would go with C. This, again, is a very clever way, nature settled on this. These are these random things that happen, and they happen to stick because they work. And what are they doing while they work? They're preserving the information that we want. So what does that allow us to basically do? We can use one strand and then create another, and it would then be identical to another strand. That's a bit of a spoiler for the replication bit that we're going to get into next. And I also mentioned the linkages in between. These are things that I want you, when you see them, to be comfortable. It's really important we're going to be doing these questions both in practice and on the real thing, which is, again, a very different test, but you could use this same kind of testing approach in class to be comfortable with what you're seeing. If you see something like that phosphodiester bond or something like that, that may not be labeled, but there's an arrow pointing to it or something like that on one of your tests from your teacher or something, you don't want to freak out and then not know. So that's why I'm putting in a little bit more information to provide you some context and set the scene. Okay, and I mentioned these words in passing, and some of them were on the slide. A gene is essentially just a portion of the DNA that is responsible for getting transcribed into RNA, talk about that, and then translated into protein, which is really the stuff that's doing all the heavy lifting. A genome is all of those genes for a species. And like I said, we've got about 20,000. Chromosomes are how those bits of the genome are divided up. I've been talking a lot about humans, or eukaryotes. You're also going to want to draw some distinction between eukaryotes and prokaryotes. Prokaryotes don't have a nucleus. They are basically the bacteria in the world. E. coli is probably a very common one that you've gone over in class and discussed. How does that differ? We were talking about the packaging of DNA and everything. Well, the DNA, let me go ahead and see if I can also make this, can I get this to, once again, I did not change my name here for PM classes. I'm poor at apparently remembering to do that, but I'm hoping that when I pause a share like this, that you then see the video of just me. But you're going to want to make distinctions between prokaryotes and eukaryotes. Prokaryotes that don't have a nucleus and are bacteria and far more simplistic, they're inherently going to have differences because they don't have the same complexity. They don't have the same needs as eukaryotes. So they actually have one chromosome, and it's in a circle. It's a circular genome called a plasmid, and a number of things are going to be different for them in terms of replication and how their genes are organized in the genome and everything. But I just wanted to draw that difference. A lot of these processes are going to be tailored towards eukaryotes. Prokaryotes, one chromosome in a circle, not that whole, condensed version down into the chromosome that has the typical X structure. I also mentioned the DNA and the histone proteins, and then the different kinds of chromatin. Euchromatin is active, heterochromatin is inactive. So moving on to DNA replication, one of the several molecular bioprocesses we're going to have to know here. Now, this is our double-strand DNA. Notice this is a proper helix because it has a major groove and a minor groove. I am such a nerd. What we're going to actually end up having is have this get pulled apart, and then you have these other strands coming in here like this, and then those continue along until we have two new identical strands of DNA. Now, this means that it is semi-conservative. So not conservative as in it's all of this and then all a new one. Imagine two strands where one was all blue and one was all gold. That's not how it works. It's also not just basically a combination of the two where it's some blue, some gold, some blue, some gold down each and every strand. It's one blue strand, one gold strand, or one parent strand, one new strand. That is also another important detail that is prime fodder for some of the AP questions. So that we call semi-conservative, where one strand is at least conservative. on each replication event. How does this replication even happen? This is a boiled down image of what actually happens here. And I did that on purpose because I wanted to highlight only the proteins that you actually need to see. And by I did this, I do not mean that at all. I found it and I liked it and I put it in my slide. Thank you, Smith from 2019, who produced this image. But essentially, we've got that double helix. And you know, also there's some interesting experiments if you're interested in how we determine the semi-conservative model. Again, throw out some questions if you've got any, so that way I can let you know whether or not it's par for the course here. But we need to unzip that, as we saw in the previous image. So how does that happen? Well, there's an enzyme called helicase. Enzymes tend to end in A's. Again, just a naming convention that we have and we all kind of agreed upon and we move forward. Kind of like how biologists make funky shapes and then label them, whereas chemists would actually care about what molecules and chemicals are in there. Everybody kind of has their level of magnification that they can bother to go to. So a helicase is the enzyme responsible for unzipping this. But the problem with that is that, and I don't actually have a shoelace available. I can use these. So I'm going to go ahead and stop share. And now I know I am front and center on my camera. So, okay, double helix, helicase, unraveling. Now, if I were to try to wind this up even tighter, so I'm just twisting, I'm trying to actually twist here, twisting, twisting, twisting. You see how it's buckling? This is a lot easier with a shoelace. Maybe on this side I can do it. It's going to end up buckling and forming those kinks. Again, this works so much better when you don't have lotion on your hands from washing them 5,000 times a day. But they end up forming those kinks, and it basically gets tighter and tighter and harder to both twist or to unravel. You don't, when you're unraveling it in order to unzip it, you don't want those kinks to simply happen in the opposite direction. So what do we do for that? How does the body basically produce an enzyme to make what we need to happen faster? Let's go ahead and do that. Topoisomerase. How the heck do you remember the name of that enzyme? Well, topo is kind of like topography or the lay of the land. And then isomer, just like in chemistry, an isomer is the exact same formula, but in a different orientation. So what this is doing is it's maintaining the DNA itself. It's not changing where those bases are or anything like that, but it's fixing the topography to kind of relax it as it goes. So helicase is doing the unzipping, and topoisomerase is picking up after it in order to relax it and get the kinks out. Okay, so then how do I actually make the new strand? Well, you notice that we have two new strands we're making here. One we call the leading strand, one we call the lagging strand. Why are they called that? Remember the directionality that I talked about? That's once again coming up here. So as you unzip and you have a 5' and 3' end, you have the opposite ends in the opposite orientation on the other strand. Enzymes, in order to be super specific, are going to be picky. Essentially, in order to be specific, they're specific. That really explained a lot. But they're going to be picky about what they can bind to and what they can act on. They can only add nucleotides or new bases in order to make the new strand at the 3' end. So the polymerase, which I didn't really talk about here, polymer A's, DNA polymerase. So a polymerase is going to create a polymer. It's going to add monomers of these bases on one after another. And what is it adding? DNA. So it's a DNA polymerase. Hint, there's also an RNA polymerase. It can only add on the 3' end. So on this, when this opens up, and if this, let me follow this along here, follow the green line. Okay, here. If this is the 5' end, should I have labeled it? Absolutely, but I didn't. If this is the 5' end, and you come along here, and then this is the 3' end, well, then this is also the 3' end as you unzip it. And so the polymerase is able to add on bases here, one after another. That means it can lead the way and just continue as the helicase unzips. No issue. But the lagging strand, not so lucky. So this now is the 5' end, and that's the 3' end. But it's the same enzyme. We don't have a 5' polymerase and a 3' polymerase because we solved the problem a different way, essentially. But the DNA polymerase, putting things on the 3' end, is basically going to need something that looks like the 3' end. So the primase comes in and it puts on what's called an RNA primer. It's not one base, it's usually a few. Smith did the best he could with this image. But when that RNA primer is put on, the DNA polymerase can recognize the 3' end of that and then add on additional bases. But the problem is that as this continues to unzip, it's going to have to continually skip. It's going to like attach, add some bases, and then it's going to unzip more. It's going to have to attach, add some bases, and then it's going to unzip more. It's going to keep doing that. And so it's what it's laying down, those segments or fragments of DNA that are being added are called Okazaki fragments, likely named after someone with the last name Okazaki. That's all I got for history on that. But as it does this, it's going to basically take longer, which is why we call it the lagging strand. It keeps waiting for more to be unzipped so that when it adds to its 3' end, it's looking for more 3' end to be available. So what do we have here? We have a helicase doing the unzipping, a topoisomerase picking up after that and relaxing the DNA and fixing it on the back end, just also relaxing it in order for it to be unzipped because we have to uncoil it. And that requires sometimes cutting and then putting it back together. I'll go through that on our vocab slide after this. And then the primase coming in, putting a primer, it's an RNA primer, and then the polymerase putting down the bases. Now, if we also have a bunch of these Okazaki fragments, there's one other enzyme that you're going to need to know for this. Ligase, as in to ligate or tape together or adhere together. That's got to come through and glue them all together, basically all those fragments. And we got to change all those RNA primers into DNA. So we have an enzyme that comes through and does that. Okay, so I'm going to go to the next slide. I'm expecting some questions. There's a lot going on with this process, but it's important that you know it. So semi-conservative, helicase unwinds the DNA double helix. Topoisomerase prevents tangling. Polymerase is the thing doing all the real work, making the new DNA strand. RNA primase puts on those short RNA primers. Okazaki fragments are those little bits on the lagging strand. Ligase joins them together. One other thing. If I were to use the same chord, which essentially failed me earlier, also apologize if the resolution on this is absolutely terrible. All of my equipment is very old. But imagine moving along this strand and getting towards the end in order to try to replicate the DNA. But the protein itself is in the way. It takes up space on the DNA. So what about that last bit of DNA at the end? We call those telomeres. You know how shoelaces have those little, this is why I wanted a shoelace. You know how shoelaces have those little plastic caps at the end? Those are actually called aglets. Again, fun fact, no one actually cares. But telomeres are important. Telomeres are a bit of DNA at the end of our strands that we fully expect to kind of lose after each replication event, just a little bit of it. So those get shorter over time. The good news is nature realized because that keeps happening, don't put anything important down there. Just put some non-essential DNA at the end to fill a space. And there are enzymes called telomerase that kind of fixes that up and adds a little bit so that they don't get ridiculously short really fast. But you don't really need to know any of that. It's just a fun fact. So the end here would not be able to get copied, and we would call that the telomere. I had a great question just come in. I was fully anticipating this, so I'm really glad someone mentioned it. Are we going to have to know the difference between DNA polymerase 1 and 3? According to College Board and what is covered, no. Now, if you have an interest in that, because it's not going to be necessary to know for the test per se, if you're interested in that, go ahead and shoot an email to classes@prepmatters.com, and I will happily explore that with you. It's interesting in terms of which one, how it's substrated that enzyme, what it works on, differs, but no, you don't need to, and that's why I didn't put that detail on the slide. DNA, double-stranded, stable, able to preserve our code that leads to our ability to then know how we make something and preserve that recipe for future generations, like your great-grandmother passing down the recipe box. Awesome. How do I actually, though, get to the idea of protein, which were those big macromolecules that I've always keep mentioning on machines and doing all the work? We have this thing in biology called the central dogma. That essentially tells us that DNA is transcribed, let me do this from the right camera side here, DNA is transcribed into RNA, and that's translated into protein. RNA is always our middleman because it has a slightly different structure, and because this also feeds into that whole idea of RNA probably came first. If RNA came first and led to proteins, then it would make sense that as we're trying to make something more stable, it would still pass through that same thing. Nature has zero interest in extending herself to do something that doesn't have a point. If it doesn't work, or it's not the most efficient thing to come along, it doesn't stick around. So we have DNA transcribed to RNA, translated to protein, and then making more DNA is replication. Any idea what this here with the red arrow and the star is? That is actually perhaps one of the most interesting things given everything going on in the world right now. This is reverse transcription, and I'm going to have a slide that shows that. Now, we'll talk about that in a moment when I have the other ones. Where, apologies if you hear anything in the background, this is a pandemic, we all are home, and I have a 2-year-old. So where does DNA transcription into RNA occur? Let's try that question on. And also, where does RNA translation occur into protein? You can think about how structure also ends up leading to function, and we could think about how bacteria were simpler and started off before the higher order eukaryotes like us, and how they didn't have a nucleus, but they were still making proteins with their RNA. So if there's something that bacteria don't have, there's still the bit that they do have, and then there's when all of a sudden things got more advanced for us in eukaryotes where we do have that other extra thing. So the answer to these would be yes. Someone got it in right before I clicked. Transcription happens in the nucleus, and translation, I wasn't quite thinking ribosomes. That is very specific, but I was thinking inside the cell, the cytoplasm. So there isn't a nucleus in bacteria, so their translation, the event there, the proteins that are responsible, the ribosomes, which the student mentioned, they're in the cytoplasm. They're in the general kind of just, auditorium of the cell. The principal's office is where the actual, the nucleus, where the DNA is, where it's being transcribed into the RNA, and then some other things have to happen before that RNA comes out. But those are also two very key points that we want to make sure we remember. Now, this reverse transcription, what's so interesting about that is that we come up with rules and we kind of discover new things, and then we have to change them up because science is alive. It's a breathing thing, and it's constantly able to be edited, whether it's don't wear a mask outside or wear a mask outside. In this case, I wanted to bring it up because I think that if we all knew a little bit more about this stuff, I think it's great that you guys are taking AP Bio. I think it's great that you're here and you're trying to get whatever sources you can for some review. If we all knew a little bit more about this, maybe we'd all be able to take care of ourselves and each other a little bit better in the real world too. The reason I say that is because the coronavirus, SARS-CoV-2, another detail that no one else cares about unless you're a virologist like me, is an RNA-based virus. So it is actually doing this reverse transcription into DNA and then going back to RNA and making its proteins. And I just thought that would be an interesting thing to include. Okay, so transcription, the other process that I wanted to discuss here. How do we get from DNA to RNA? Again, a lot less detail because a lot of similar players. For instance, I think you understand this is going to need a helicase to unwind. This is going to need a topoisomerase to fix that. There will not be a need for primase because we are making RNA. There is also no need for, and I'll talk about the question that I addressed earlier, there is no need for Okazaki fragments or ligase or a lagging strand because we're only going to use one strand of DNA. Now, since DNA is more stable and it likes to be double-stranded, there was a really good question that came up about single-stranded binding proteins, or SSBPs, you may see them as an acronym, and then not really explored because you don't actually need to know them for the test. But essentially, a lot of times when something that is less than ideal, like single-stranded DNA, which is a thing and does exist, needs to be stabilized, there are proteins that kind of do that. Just like the histones allow the DNA to wrap around it, make nucleosomes, and then pack it together and make our chromatin into our chromosomes, we have single-stranded binding proteins that bind the DNA on the single strand. I'm pointing at the screen as though you can see where I'm pointing. Here with my cursor in order to basically stabilize that. But this big purple bubble here, no, it is not necessarily this large in comparison to the DNA and RNA, likely because it's a giant protein. It's not necessarily much bigger than the DNA polymerase. But I did want to point out RNA polymerase because we are making or putting down RNA. So 2 strands. How do we choose which one? Doesn't really matter. It does in terms of knowing that the name of the one you chose is the template strand. A template, just like for your essays or your outlines or anything like that, is basically the thing that you're basing something off of. So the RNA strand is based off of that. Still the same complementary base pairing, except, pop quiz, which base is not going to be laid down by RNA and is going to be substituted for something else. It's not going to put down thymine because that is a DNA base. There it is, right there. Thank you, Maddie. It's going to put down uracil. So these nucleotides are kind of just hanging out, and they're there for the RNA polymerase to take and then lay down. This other strand that is not being used is called the coding strand, because if the RNA is getting put down and it's complementary to the DNA, then it's going to have the same sequence as this strand. So the light blue one here is our template. We're basing our new RNA off of it, and then our other dark one is our coding strand, and that's going to have the same base sequence as our RNA. Now, looking at the clock, I got to move along a little bit faster here. Okay, now we've made my RNA, and I talked about how that was in the nucleus, and then we got the cytoplasm. Does it just go out? No, it needs to put its clothes on in order to head out the door. So what does that mean? Well, DNA is not as compact as RNA in terms of getting right down to business in terms of genes. There's a bunch of stuff, just like that telomeric DNA at the end. There's a bunch of different parts of the DNA that don't actually go on to be part of the protein. Those parts are called introns. So exons are the things that are expressed, and introns interrupt those. There are a million different ways you could make exon sound like the part that you're going to have cut out and introns be left in. Just exon expressed, intron interrupted. So exons are the bits that code for the genes and introns do not. We think that introns are probably doing something like helping organize all of these different genes in place. But either way, we have this thing called pre-mRNA. There's actually a bunch of different kinds of RNA, and I will mention those in a moment. But those then have to get spliced and packaged together to make our mature mRNA here. All the introns are removed by a protein called a spliceosome. Don't really need to worry about that either. Do we put anything on either end here? We have to put on a 5' cap and a poly-A tail. 2 details that we definitely have to know too. These are kind of like a signal that the RNA is then, the mRNA is then ready to leave. What does the M stand for? Messenger RNA. It's the messenger. It's not the actual thing, not the protein, but it's messaging as to here's what should be made. There are other kinds of RNA, ribosomal RNA. Oh my goodness, so many different kinds. tRNA, we're going to talk about those two in a moment. And then a bunch of different kinds of RNAi or RNA interference. Small nuclear RNAs, microRNAs, silencing RNAs. Another reason why RNA likely started this whole shebang off is because they have all these different possibilities. They are reactive, they're catalytic. They themselves can act on other things and tag other things so that proteins act on those bits that they tagged. So in this case, we're just talking about mRNA, which then leaves, when it's mature like this, with the 5' cap and the poly A tail, leaves the nucleus to go to the cytoplasm and get translated. So I mentioned ribosomal RNA and tRNA just a second ago. Let's see, we got a quick question here I saw. Is the RNA world hypothesis accepted by the scientific community? For the most part, yes. This was, I would say, a number of years since I was on the other side of this in the classroom, but we were still learning that even then by my professor Jeff Buca at Hopkins, there's so many different things about RNA and how we approach everything, but I want to focus in on this and move it along. But yeah, that is how we actually think things transpired in terms of chronology. Okay, translating RNA. How do we do this? This big blue thing here is the ribosome. That's actually both RNA and protein mixed together. These do exist in prokaryotes because they're in the cytoplasm and they're responsible for making proteins, which is so important. It's the way everything gets done. So how does that actually work? They aren't labeled on here because it's really hard to find simple images that aren't super complex and therefore going to confuse everybody. But there are 3 sites, A, P, and E. But the A site, it stands for aminoacyl. And if you remember directionality, if you go left to right on an amino acid and you look at where the peptide bond connects them, you go from the amino end to the carboxyl end, and then there's a peptide bond to the next amino end. So you can think of the amino being the first, being on the left if you were to read English, left to right. So you don't need to remember that. You're mostly just going to be referencing them as A, P, and E. So remember A for attachment, right? And then P is actually the peptidyl site, or where the peptide bond is formed. And you could just consider that the processing, like where's all the actual action happening. And then E for exit, and where the tRNA gets kicked off. So the tRNA is this really weird looking RNA here that's all curved. Again, so many different things that RNA can do. This here, as it's pointed out, is the anticodon. It would be complementary to the codon, or the triplet of RNA bases that move along. I would love to explore this more in terms of how we got the code and why it's a triplet, and why it's not 2 amino acids, 2 bases or 4 bases. We just don't have the time. But if you do have those questions, go ahead, classes@prepmanners.com or hashtag PMTV on social media. I'm at Mental Markets, MENTALMARKETS, on Twitter and Instagram. Throw your questions wherever you want. We're going to see them if you throw that hashtag on there, and then I'll be able to answer those. But the codon is what's going to end up telling us what the amino acid is, and the anticodon basically associates with that. And each tRNA has a different anticodon, and then a different amino acid associated with that anticodon on the other end. Like this guy here, UGC, was this amino acid. And ACG was in the RNA as it moved in this fashion. Again, it enters here, then goes here, then goes here, and continues on. So you can see here, this line right here, the first amino acid, and the second amino acid, and the third amino acid, and the fourth amino acid, that kind of thing. So as this progresses this way, the tRNA binds here, and then this amino acid is hanging out on the end of the tRNA, moves to the next site, and the ribosome and those RNAs inside allow it to make that bond between them, and then it's done its job. It no longer has this amino acid attached, so bye-bye. that goes to the exit site. And then that gets kicked off. So translation, the last bit of the central dogma. We had DNA to RNA to protein. This would now be our polypeptide chain. Okay, now, again, we got a bit to go through here. We're going to go through the lac operon and the trip operon faster than you would in class. Well, everything's faster than you would in class, but even faster than I'd like now. Lac operon, what the heck is it? We've got our polypeptide chains. We've got our RNA that told us about that, and our DNA that basically told us what the RNA was going to be. We want to express a certain gene. How do we know in all these genes, like even in a bacteria, it's got a ton of genes. In a bacterium, it has a ton of genes. How do we know which ones to turn on and off? Well, sometimes we want something that is called an inducible operon. An operon is essentially a gene in prokaryotes. It's a segment of the DNA that has portions that are called a promoter, an operator, and then all the various genes. Promoter and operator, those are docking sites. That's like the scaffolding. That's the infrastructure to the actual gene. They're not translated. They're not made into proteins or transcribed, sorry, made into RNA or then translated into protein. But that operator is where what we have here, a repressor can bind. And if that's there, well, then a different protein, the RNA polymerase, cannot bind. And it can't continue on and make the, transcribe the genes into RNA and make them into protein. Well, the lac operon is to make lactase. Now, why would I want to make lactase? Well, lactase is a sugar. I'm sorry, lactase is the enzyme that breaks down sugar. Lactase is an enzyme that breaks down the sugar lactose, a sugar in milk. When someone's lactose intolerant or something like that, they don't get along with the sugar in milk, and that's why you have to have different dairy products. But typically, we want glucose. Glucose is that sugar that we talked about last time, one of the saccharides. That's the really good stuff. But if we can't come across that in the wild and lactose is available, bacteria say, okay, that's fine, like spoiled milk. They obviously like it. That's bacteria growing in the milk. So how does that actually happen? Well, they don't always need the enzyme that can break it down. And that's kind of taxing to just have a bunch of stuff around that you don't need. So in order to Marie Kondo the bacteria here and just only keep stuff that sparks joy, that's a dated reference at this point, it's going to only make the enzyme that it needs when the lactose is present. So the lactose can come in and then bind to the repressor, which causes a conformational change. Again, structure and function, another thing popping up here for a theme throughout all of our units to connect them. And then that pops off and it allows the RNA polymerase to jump on and then transcribe that gene. So that is inducible. If lactose is present, then the repressor will come off and will go through. Now, what if we wanted to repress something instead? So we were just talking about making something only when it's needed. Well, the same can be said here. We still only want to make things when they're needed. So the trip operon, which is for tryptophan, an amino acid, what do we do? Like, do we want to just keep making that a bunch of times? No. There's a regulatory gene called the trip repressor that gets transcribed, translated, and then made into this protein. That, as you can tell from the shapes here, would bind the operator here and prevent the polymerase from being able to go. When would that happen? Well, see how this has kind of a bend to it? Well, if this little guy right here, kind of like lactose sat in the repressor and caused a conformational change and then come off of the operator, tryptophan, if it were present, would bind here and then cause a conformational change so that this could bind here. The only images that I could find that demonstrated all that had a bunch of stuff on there. And I wanted you to focus on just what you needed to know. You're undoubtedly going to come across this in countless different materials that you have to prepare. But if tryptophan is present, then it would bind here, cause this to change shape, bind here, and then you wouldn't make genes that would lead to, you wouldn't transcribe the genes that would lead to making tryptophan or tryptophan synthesis. If you don't have tryptophan, then This guy isn't going to be able to be the right shape and size to bind here, and the RNA polymerase will come through and transcribe the genes in order to make tryptophan. So what I queued up here was a question as an example of something that involved the topics that we had. This is question number 4 from the 2016 AP Bio test. Some changes that are occurring here. Go to the College Board website, go to Prep Matters website, and you'll find links that describe changes that are happening to these tests. It's going to be that 45-minute exam. It's only going to be 2 free responses. The first one's going to take 25 minutes. The second one's going to take 15 minutes. That's actually only 40 minutes. I don't know why they say 45 then, but you're only going to be able to do the first one and then submit it, and then you do the second one. But I still wanted you to practice a lot of the components that I know are going to be standardized, like these action verbs that are bolded in here. So let's say you're given these DNA strands in this image here with the introns in white, exons in black. There's your primary transcript or your pre-mRNA, and then your mature mRNA, and then made into protein. So this is the process of expressing some gene. We know it's a eukaryote even if they didn't tell us, because that white area is going to be the nucleus. So the primary transcript in the figure is 15 kilobases long, but the mature mRNA is 7 kilobases in length. Describe the modifications that most likely resulted in the 8 kilobyte difference, kilobase difference, sorry, in length of the mature mRNA. Identify in your response the location in the cell where the change occurs. This is why I cover this stuff in my slides. Second part would be predict the length of the mature gene X mRNA. If the full length gene is introduced and expressed in prokaryotic cells, justify your prediction. And this is why I talk about differences between prokaryotes and eukaryotes. So I'm going to give you a little bit of time to think about that. And then I'm going to show you the scoring guidelines about what's acceptable. So go ahead and give that some thought. Okay, so we have describe, which I talked about last week. I'm going to provide a little bit more detail, but identify, just point things out. Predict, okay, kind of describe in a note what's going to happen in the situation, and then justify, you better have reasons for it. So describe is literally just, you go from this to this. We've removed the introns. You only get 1 point, so you could label it as RNA processing and then mention that that includes the removal of introns, or you can just say that the introns represented 8 kilobases of the 15 kilobase primary transcript. And then identify where this happens, the nucleus. The mature mRNA is out here, but it actually starts here. They didn't actually even require you to talk about the 5' cap or the poly A tail. Other times they do. And then what about the prediction? Well, what would it be? Predict the length of the mature gene X mRNA if the full length gene is introduced and expressed in a prokaryotic cell. So if we took that same DNA, we put it in a bacterium, and then it was expressed there, no nucleus, none of the bells and whistles eukaryotes bring to the table, how would it differ? It would be the full length primary transcript, longer than the mature mRNA that we have here in eukaryotes. Why? Because the mRNA processing doesn't occur in prokaryotes. There is no nucleus versus cytoplasm here. There is no exit that then triggers and signals for the mRNA to be able to leave the nucleus and enter the cytoplasm. So it's just going to stay the way it is and jump right into that ribosome. Everything is done in a simpler fashion. It's a bit more wham bam and then move on. So, okay, that was one of the questions I want to cover. I know they went over that much quicker. I apologize for the rate at which we have to cover all this material. One other thing I wanted to mention was, if you did want more practice, go ahead to the test for 2017. These are again on AP Central on College Board's website. They're freely available. Free response question number 3. That's going to talk more about that triplet code and amino acids and everything. And if you wanted to complete that and then you had questions about it, classes@prepmatters.com. # PMTV. Okay, thank you so much for sticking around. I hope this was enjoyable, informative, a bit more engaging. I'm going to keep trying to bring as much of a show as I can to make it interesting and keep your attention. Please send us any questions. I do love them and kind of live off of them. And until next week, we're going to talk a little bit more about the genetics and it connecting into the cells for inheritance, meiosis, Mendelian genetics, all that kind of stuff. Be well, stay safe, study hard, and thank you once again.